My name is Maria Croyle, Chief Scientific Officer and co-founder of Gerada, a company that aims to solve key distribution and delivery problems for vaccines and biological drugs. In my laboratory at the University of Texas at Austin, we've developed a method by which we embed a variety of biological drugs within a thin film matrix that protects them from extreme heat, cold, and other environmental stressors. You can see here what the actual final product thin film looks like, and you can also see here virus particles embedded within the thin film through electron microscopy. We have identified a novel compound that surrounds the biological agent in the liquid form, shown here, that protects the biological agent. And as the film is formed, it actually coats it to confer stability. We have done this with a variety of agents and use a simple manufacturing process. We have a strong patent portfolio and have shown that vaccines stabilized in the film can be given in a needle-free manner. There are a lot of stops from the time a product leaves a manufacturing site until it reaches the end user. These stops provide many opportunities for product loss due to improper handling and storage. We saw this firsthand during the COVID-19 pandemic with healthcare workers trapped in a snowstorm, rushing to give vaccines that would spoil if they weren't used immediately. The vaccine and biological drug market is projected to be worth $441 billion in the year 2024. Approximately 10% of this revenue will be lost by the biopharmaceutical industry due to improper handling and storage of their products. Half of this loss will be represented in replacing the actual lost product, and the remaining half trying to figure out what exactly went wrong and correcting errors. If one wanted to give 350,000 doses of the current BCG vaccine, we would need a space equivalent to the size of a US football field, and it would need to be located somewhere very cold, let's say Wisconsin, in order to keep the vaccine stable. The same number of doses embedded within our thin film matrix would have the size equivalent of a ream of paper and weigh approximately 1 pound, and let's say 20 pounds with additional packaging. No refrigeration would be needed in this case, as we have data demonstrating that an Ebola vaccine embedded within our film is stable for at least 3 months when stored at room temperature. We have all received injections before in our lives, and we can agree, I believe, that they are an unpleasant experience. They are also a significant reason for non-compliance and create a lot of biohazardous waste. Our technology can potentially eliminate both those issues. We have shown that an influenza vaccine embedded within the film and given under the tongue or in the upper cheek of mice has induced an immune response similar to that of intramuscular injection, and that's shown here in the left panel. We've also shown that an Ebola vaccine embedded in film that was dissolved in saline and given as a nasal spray also protected primates from a lethal dose of Ebola. There are a few companies that also aim to create thermostable biological products. These technologies rely on some form or variation of freeze drying, with the end product being a dry powder. It is important to realize that, to our knowledge, Gerada is the only company that holds a rich data set and patent portfolio for biologics stabilized within a thin film matrix. We have consulted with several regulatory consultants and determined that the thin film matrix is considered an excipient by the US FDA, and we will perform toxicity studies on our novel reagent and on the complete film formulation for each anticipated route of administration, oral being the most obvious, intranasal, and injection as well. These studies can only need to be done once, and then we can compile the data in a drug master file that we can submit to the FDA so that each of our clients and people interested in licensing our technology can refer to when they submit their own investigational applications. So they don't have to reinvent the wheel every single time if they're interested in using our technology. We plan to have a pilot production line for the films in place by the end of 2024. Our toxicity studies will be complete by mid-2023, with filing of this DMF with the FDA complete in 2024. Milestones that we've had to date are that we have a very solid client base. We have 7 active or complete sponsored research agreements with companies spanning the range of gene therapy, vaccines, and animal health. At this time last year, we actually had an offer of acquisition by a global CDMO, but the terms just weren't right. We also received funding from the NIH to develop a program looking at novel adjuvants for oral immunization. We are part of the BARDA Blue Knight program, and we've identified a manufacturer of pilot equipment within the last 6 months. We are at the end of a seed round, and with this money, we're putting it towards pilot line development, our toxicity and bioequivalence studies, and establishing a GMP supply chain for all the reagents needed to make our film. We are also now opening a Series A round to support pilot manufacturing, R&D infrastructure, our regulatory submission, and really expanding our production line so we can enter clinical testing with partners in the relatively near future. I'd like to introduce you to our team. There's myself, who's the founder and chief scientific officer. I have over 25 years of drug delivery and formulation experience. This has allowed me to serve as a consultant for a variety of pharmaceutical and bioprocessing companies. My laboratory has worked with the USP and the FDA to establish standards in the gene therapy and bioprocessing space. Our CEO and third co-founder, Sheila McHale and Jude Samalski, have an extensive record of taking disruptive technologies from the lab bench to the commercial scale and creatively designing exit strategies for each of these that are quite beneficial. Chris Pavlos, our director of R&D, and Megan Livingston, our director of business development, both have experience in material sciences, in experience in the industry, and really are a resource of endless energy that really helps us get this project moving forward. What we're asking for today is we're closing a round of seed funding, but there is still some space available for people to contribute to that. We are running a Series A, as I've already talked about. We're looking for strategic partnerships with companies potentially to validate our large-scale production process, because this is a relatively new and disruptive technology. And we're looking for a vaccine and/or biologic supply for bioequivalent studies, but very importantly for scale-up manufacturing, which will require a significant amount of material. So again, just to summarize, our technology eliminates the reliance on the cold chain, increases manufacturing efficiency, can offer needle-free administration, and is cost-effective and resource-sparing. Thank you for your attention. I'd be happy to answer any questions that you might have.
Fantastic. Thank you, Dr. Croyle. It looks like, Jeff, you have your hand up.
Yeah, so this is awesome. You did not mention that you've already tested this on mRNA vaccines, but it's low-hanging fruit. I'm sure you've thought of it if you don't even have data. And the second question, kind of related to that, something else that we have to refrigerate, although not as much as we used to, is insulin. You did note that you can stabilize proteins. Given that, have you looked if you could deliver insulin subbuccal and store it at room temperature? It would be a pretty big deal in a 21 billion, if I'm correct, size market.
That's a very good question. So I'll tackle your first question first, which was about lipid nanoparticles and mRNA. We actually have an active contract right now where we're working with a partner in developing a platform for them to exactly do that. And we haven't necessarily considered insulin quite yet. Only because our expertise is really in the viral and vaccine market, but that is something that's intriguing that we could potentially consider as well.
Gladys?
Thank you very much for the presentation, Maria. Nice job. Just as, like you said, you're raising money for production. Have you kind of at least done at least a minimal kind of cost of goods analysis as you're kind of planning on raising money? And because I know you're going to perhaps have to do several testing around that. So have you at least kind of outlined some costs to do that?
So we have developed a budget for that, and we know what those expenses will be for a pilot line. And like I said, we're working right now. We've initiated a contract with a contact lens manufacturer, believe it or not, to actually adapt their technology to what we're looking at. And this has really been one of the things that's been interesting to work with potential partners. Is it's realized as we talk to them, everybody understands lyophilization and freeze drying, and they're like, so that's what you do. And I remember, like, that's not it at all.
That's not it. Yeah.
And it's very disruptive. So talking to people that are really within the pharmaceutical industry has been tough, but it's interesting that the contact lens people kind of get it a little bit more, and we can really adapt what they've been doing to get something that is more amenable to our system, if that makes sense. So yes,
No, it makes sense.
We brought them on board, and we really hope to have prototypes running by the end of this year for sure.
No, that makes sense. And sometimes it's good to kind of step outside the industry, right? And do that. My recommendation would also be, there is kind of, especially due to COVID, there is a lot of investment and activity going around novel manufacturing. So there's an organization that's called Resilience to look in, and they also provide funding and are looking for disruptive ideas to optimize. So definitely look into that. Maybe that there's some things that they can do, and in addition, maybe support the pilot with some funding as well. So just some ideas. I think it's a very much needed breakthrough. So congratulations.
Oh, wow, thank you for that.
Thanks. Mitzi?
Yes, thank you for that really great presentation. I especially loved the visuals, including the vaccine journey, or the journey of a vaccine. I thought that was really a great one, as well as the visual about how space saving what you're doing is. So that was pretty impactful. A couple of disparate questions. First of all, going back to Jeff's question about mRNA, and I'm just wondering, again, given the really current need to getting vaccines globally, are you eligible, if you will, for emergency use authorization with the partner that you're working with as well? And then again, second disparate question. You may have mentioned this, but I don't remember. Do you have non-dilutive funding in terms of, and have you applied for SBIR funding?
I'll answer your second question first. Up to this point, we've either been running on NIH grants, and that's where the technology developed. This has been like a 10-year. I had a huge grant to develop a needle-free Ebola vaccine, and this is where this actually came from. And we've been running on founders money at this particular point. So, that's basically what we've done. And we've been bringing in money with these sponsored research agreements as well. I mean, this year alone, we will probably bring in about $600,000 in revenue, just working with partners who are essentially just testing our technology to see if they're ready to go to the next step, to look at manufacturing or clinical testing or things like that. To answer your first question about EMA use, this would be, great to be used with some product for that. Also, full disclosure, the first, peer-reviewed publication we had about this technology, due to the fact we did a lot of work patent protecting before we released it into the world, I guess you can say, was a week before COVID hit. And so, timing was kind of odd, but good at the same time. And so we had a lot of people interested, in doing this right away with potential vaccines, but we were a little hesitant in doing that because we just wanted to make sure that we weren't, the reason for any disruptive in getting vaccines to people. But I think we're at a position now where we could definitely do that with a variety of things should we need them again.
Absolutely. And just going back to the SBIR funding, and again, you're very far along, and so that may or may not be relevant, but if that's always a feather in your cap when investors are looking, and we love non-dilutive funding. So
thank you. We will definitely, look into that. We're actually applying for a CEPI grant as well, because they have a big campaign right now for thermostable vaccines, most again focusing on mRNA and lipid nanoparticles. So
terrific.
yes,
Thank you.
I think we have time to sneak one more question in, Jeff, if you want to go ahead.
Yeah, I'm sure you have thought of this, but the only concern I would have, this is kind of a setup to be misappropriated by somebody interested in bioterrorism. As an example, in Ebola, I am sure you've thought of it. Are there ways to address that? Because it is kind of potentially scary, yes?
I haven't really thought of it that way, I guess you could say, because, you know, most of the things that we're using are countermeasures or protective measures for that. The thing again that's really nice about it is it is very relatively carefree with respect to shipping and distribution, but, you know, I don't necessarily see how the tech, I guess you could, yeah, I guess you could see maybe that you could store things and do that way, but that's something we'll consider as we move forward for sure. Thank you.